Hello and a warm welcome to everyone. My name is K.O. Satish, Associate Vice President of Spinco Biotech, and I'll be moderating today's session. The recent COVID pandemic has brought mRNA technology out of the laboratories to commercialization. The accelerated discovery paths to mRNA-based vaccines, along with the small footprint and high productivity manufacturing, makes it an attractive proposition for a variety of challenges. Messenger RNA or mRNA is a pivotal molecule of life involved in almost all aspects of cell biology. mRNA has recently come into the focus as a potentially powerful drug class able to deliver genetic information. This webinar gives you a comprehensive overview of the current state of mRNA drug technologies, their applications, and crucial aspects relevant to mRNA-based drug discovery and development. Aptly, the topic for today's session is adapting mRNA to therapeutics. So in the next 1 hour, we are going to cover introduction to the mRNA landscape, the technologies of mRNA production, and the challenges associated with the mRNA platform, and finally, analytical approach towards quality mRNA therapeutics. So it gives me immense pleasure to welcome our panelists for today, Dr. Mona Lisa Chatterjee, Director of Sekkei Bio, Dr. Sivasa Dwarkanath, Sekkei Bio, Dr. Anand Kedkar, and Mr. Ashutosh Sheller, Assistant Manager, Shimadzu India Private Limited. Sekkei Bio is an innovation company. Sekkei means design in Japanese. So it was founded in 2017 with a simple desire to design differentiated molecules supporting efficient drug discovery. Sekkei has created over 1,000 novel insulin analogs in silico. Post-extensive optimization, selected a lead candidate, and with experimental evaluation, it has moved towards the goal of making insulin conducive for oral delivery. And this was just the beginning. Sekkei designed a potent multi-targeted non-opiate analgesic, which was another huge challenge. And in 2021, with the strength of mRNA technology, Sekkei is working on second-generation RNA technology to enhance stability, improve half-life, to convert or replace MAP therapies into mRNA therapeutics. So first, I'd like to welcome Dr. Mona Lisa Chatterjee, our first speaker. So the title is Introduction to mRNA Landscape. So Dr. Mona Lisa has around 16 years of experience working in companies like Biocon, AstraZeneca, Bill and Melinda Gates Foundation. She did her postdocs from Yale University School of Medicine. Her deepest interest continues to be in leading, participating, contributing to science that translates to better medicine and accessible to all. So over to you, Dr. Chatterjee.
Thank you so much, Satish. And I welcome you all who are joining for today's webinar. It's a pleasure to be here. I'll just take a second to share my screen. Okay, so my hope in next 15 minutes is that I'll be able to provide a high-level view of the field and the innovations that are happening in the field and the excitements that there are in the field. So RNA therapeutics, as we are all aware, are the new class of products that are based on RNA or ribonucleic acid. There are different kinds of RNA therapeutics. Messenger RNA is one of them. So messenger RNA or mRNA are, as we have been taught, are the intermediary molecule between our genetic code, that is DNA, and our functional members that are proteins. So mRNA is transcribed in the nucleus of a eukaryotic cell, transported to the cytoplasm where it is translated into proteins. Antisense RNA is an RNA that is complementary to coding RNA and used to prevent translation of already existing RNA. Similar RNA interference is brought up by small interfering RNA as well as microRNA. And RNA aptamers are short single-stranded RNA that bind to biomolecular targets, impacting their activity. So if you can see, RNA actually is the only type of RNA product that enables a desired protein to be expressed in vivo in a human body. So what COVID has done for us, it has shown that mRNA technology is well suited for rapid vaccine development and manufacturing on a global scale. So if we quickly look at the COVID vaccines, how do they work? So most COVID vaccine use a structural protein of the SARS-CoV-2, spike being the most favorite one, express it as an mRNA and then encapsulate it in lipid nanoparticles. And then it is delivered in our body as an IM injection. Once it enters our body, it is taken up by a tissue-resident cell where it is translated. I'm not sure if my cursor is visible, but it is translated, processed, and presented through MHC class 1 to a CD8 T cell. Protein can also be secreted out, which then gets taken up by antigen-presenting cells, you see on the right, and is shown. Together, it leads to a very potent B cell response. If you can see that why mRNA, the advantage is there is no risk of insertional mutagenesis. So mRNA doesn't go and integrate into our genome. It does not need to get into the nucleus. If the mRNA that is delivered is ready to translate. It is rapid, scalable, and hence very applicable for the vaccine programs. If you look at the very conventional mRNA vaccines, they have a cap, which is the translational signal. They have untranslated regions. Of the mRNAs are modified with to reduce immunogenicity. So classical mRNA vaccine development would need identification of target antigen and immunogen design, mRNA synthesis, purification, characterization. Naked mRNA is very labile and immunogenic, hence encapsulated in lipid nanoparticles. And finally, a body of work is needed for understanding stability and analytical characterization of mRNA and the lipid nanoparticle. Design parameters for vaccines include multiple features like immunogenicity and safety. However, they also have to consider multivalency, which is protection against various serotypes. For geographies geographies like ours, cost, storage, and transportation conditions, as well as raw material use, become very important. So why do we feel that the COVID vaccines suddenly were developed? They actually has been parallel evolution of the mRNA technology and its understanding, starting from when mRNA was discovered to in vitro translation and injection into a cell-free system and a preclinical system. Identification of nucleoside modified mRNA, reducing immunogenicity, and finally clinical trials. In parallel, there has been development of the liposomal technology, which started in early 60s, which today has led to the creation of current vaccines. If you look at the application landscape, divided into two different kinds of applications. The first one is where human cells, eukaryotic cells, are taken out. They are modified using a transfected in vitro transcribed mRNA, and then they are put back for a desired function. The applications range from cancer immunotherapy. For example, tumor-associated antigens are expressed via mRNAs, or chimeric antigen receptors can be expressed using mRNA. Vaccines against infectious diseases, allergy tolerization, protein replacement or supplementation therapy for genetic disorders, and then genetic engineering. If I look at the indication space, you can see there has been an explosion of vaccines for various bacterial and viral diseases, as well as the largest application you can say is in oncology. If you look at July 2021, the largest number of products in development are for tumors, followed by infectious diseases, and then metabolic, cardiovascular, and respiratory diseases. So what I'll do is I'll give you a little bit of rationale why mRNA is becoming relevant for each of these use cases. So if we look at genetic disorders, what's the rationale is to restore the deficient or defective enzyme protein with mRNA-coded functional protein in a target organ. Preclinical proof of concept has been obtained for multiple rare diseases, and the field is slowly moving towards clinical proof of concept. Very interestingly, mRNA is also entering the therapeutic monoclonal antibody field. We are all aware of the impact therapeutic monoclonal antibody has on various diseases, including oncology. However, the challenge has been the wider accessibility has been hindered due to high cost of production, as well as large manufacturing capacity that is needed. So the effort has been antibody inside the body using an mRNA. Again, preclinical proof of concept has been achieved for passive immunization against HIV, rabies, as well as in lymphoma models or cancer. There are still quite a lot of hurdles that need to be crossed before this actually becomes a reality. Shifting gears, I'll take you to the second class of use cases where white blood cells are taken out from human body. T cells are expanded and then transfected with mRNA that can express a range of things. They can express a T cell receptor, a modified chimeric antigen receptor, immune enhancers, or even enzymes that enable genetic engineering. And then they are put back into the human cells. So cancer immunotherapy observation that if you take IVT transfected dendritic cells and put it back into human body, it can then present tumor-specific antigens, which then enables a very strong immune response against the tumors. This field has advanced, and now it has been shown that even if you take naked IVT mRNA and you put it into lymph nodes, deliver it to the lymph nodes in our body, it can tumors that is specific to the patient. And the first in human testing of naked IVT is actually BioNTech is running this for melanoma. And finally, the field is again evolving to using combining self-delivering RNA with the ACT therapy. Many clinical trials in the oncology field and many different tumor types are being tested. There are no approved products yet in this field. Taking a quick look at how mRNA is impacting CAR-T therapy. When T cells engineered through mRNA electroporation has been used, it conferred enhanced safety features. And this has been attributed to the transient expression of the transfected mRNA. Shifting gears a little bit, the field is seeing innovations at a rapid pace with respect to technology, methodology, use. The IP space is very competitive and complex, with new players entering the space almost very rapidly. And I would like you to show that while I've been talking about non-replicating linear forms of mRNA, which is what we have for today's COVID vaccines. However, the field has really moved quickly to self-amplifying mRNA, which, as name suggests, can make copies of itself inside the body, or circular mRNA, which is a much more stable form of RNA. With each of these new technologies coming in, the second generation technologies, the IP landscape continues to grow in this field. So I hope I have been able to give a quick flavor of how the technology is evolving and how we can make best use of it. And over to you, Satish. Thank you so much.
Thank you so much, Dr. Monasila. Thank you for wonderfully covering the complete landscape of the mRNA therapeutics. So for all our viewers, if you have any questions, so please type in the Q&A box only in the bottom of the screen, and the panelists will answer at the end of the session. I think it's time to have a poll question now. So just to understand what is the mindset of the viewers about mRNA. So I'll just post the poll question. Yeah, so we are getting the answers. So the question is, could mRNA-based product disrupt pharmaceutical industry? So the choices are yes, no, or not sure. So we'll just wait for another 10 seconds before we close this poll. Yes. So yeah, so 49% of them feel that mRNA-based products will disrupt the pharmaceutical industry, and 37% feel no, and 15% are not sure. Yeah, any comments on this, Dr. Mona Lisa?
Yeah, thank you so much. I think I agree with the 49% saying that there is a potential, but I also agree with the 37% because this is a new technology. A lot of things have to be worked out before it becomes really a, product becomes commercial is a very different situation. So I do agree with both segments. We have to just see how this gets applied and how can it really become applicable to everyone in the world, like all geographies, or will it get restricted to very few geographies? I think it's just, we have to wait and see. Yeah. But it's an exciting time. I will Absolutely. Not take away that.
Yeah. Yes, so going forward, I once again repeat, please all of you just type it in the Q&A box only, because if you type it in the chat box, we might be missing your questions. Please type it in the Q&A box so that the panelists will answer your questions. So going forward to our next speaker, a quick introduction of Dr. Srivastava Dwarkanath. So he did his MS from the University of Essex and his postdoc from Max Planck Institute. He has worked as a postdoc research in IIC and currently is a part of Sekkei Bio as a scientist working on mRNAs. So he too believes in the vision of Sekkei to improve human health by developing drugs and vaccines that are designed for all. Over to you, Dr. Srivastava.
Thank you, Mr. Satish, for such a kind introduction. I'd like to welcome you all for this presentation, which is titled Technologies for mRNA Production. In this presentation, I'll be covering, okay, let's just wait for the slides to change. Okay, in this presentation, I'll be covering a process called as in vitro transcription, with which we make mRNA. Then I'll be talking about the structural elements which are important for mRNA functionality. Then we'll talk about how these structural elements can be incorporated into your DNA template elements. Finally, once your DNA template is ready, we'll see how mRNA can be produced and purified. At the very end, I would like to talk about some of the exciting work which we are doing at Sekkei currently. To begin with, in vitro transcription or in vitro runoff transcription is a process of making mRNA. Basically, it's a single pot reaction in which we have a DNA template, our RNA polymerase, NTPs, and other cofactors like magnesium ions and spermidine in this pot. Since the DNA template contains a promoter region, the RNA polymerase recognizes it and it comes and binds to it. So I hope all of you can see the cursor pointing towards it. Upon binding, the RNA polymerase sort of destabilizes the promoter region and splits the two DNA strands. And the template strand of the double-stranded DNA acts as a sort of original copy for synthesis of mRNA. Once the RNA polymerase reaches the end of the DNA, it just falls off and you get the mature mRNA being formed. As you can see in this cartoon, the mRNA is highly dependent on the DNA template. And many times, structural elements which are required for the stability of mRNA has to be incorporated beforehand in the DNA template itself. So what are the key structural elements that are required for an mRNA? Well, they are the open reading frame, which are flanked by the 5' and the 3' UTRs. And finally, at the 5' end, we have a 5' cap. and at the 3' end, a poly A tail. The open reading frame is, as the name suggests, is the region where our protein of interest will be encoded. This region has to be sequence modified in such a way that we get better expression. The 5' UTRs and the 3' UTRs are the regulatory elements which help in improving our translational efficiency, whereas the 5' cap and the poly A tail are required for the stability of mRNA and initiation of the translation. In the coming slides, let's just go one by one and get deeper into what these structural elements actually do. So let's begin with the untranslated regions or the UTRs. The 5' and the 3' UTRs, as I already mentioned, are regulatory sequence elements that have an effect on the mRNA secondary structure, and thereby they have an effect on the stability and degradation of mRNA. The 5' UTR also contains elements which help in ribosome recognition, as well as elements which help in interaction with translation machinery, thereby having an influence on the translational efficiency of the mRNA itself. The 5' UTR can be modified in such a way that we can improve the mRNA stability and translation accuracy. And this can be done by avoiding presence of start codons or non-canonical start codons in the 5' UTR, because of which you get proteins which are not of expected sequence. We can also improve the stability by avoiding many stable secondary structures, because of which the ribosome gets stalled and falls off. And both of this can be achieved by reducing the length of the 5' UTRs and removing elements which are not required. The commonly used 5' and 3' UTRs right now are derived from the alpha and beta globins. Moving on to the 5' cap, a functional 5' cap is required to shield the RNA from the 5' end from attack from exonucleases. It also is a signal for binding to the translation initiation complex and thereby initiating the mRNA translation. Another important function that the 5' cap sort of has is to differentiate between self and non-self RNA molecules. So many viral molecules lack the 5' cap, and because of which when they enter the cell, they get degraded instantly. Most mRNAs are capped either by post-translational enzymatic capping. This is majorly done using the Vecina capping system. And as the name suggests, this is a capping mechanism which is done after the transcriptional reaction is completed. So if you look at the cartoon at the right-hand panel, the A part of the panel describes the post-transcriptional capping very efficiently. So once the transcription reaction is done, the 5' phosphate is removed by phosphatase, and then using a glucosyl transferase, a GTP molecule is added, and using methyl transferases, the cap-like structure is formed at the 5' end of the mRNA. And all this happens after the transcription reaction has ended. Another approach of getting a 5' cap incorporated is the co-transcriptional capping, or as the name suggests, transcription capping while the transcription reaction happens. Commonly used co-transcriptional caps are the trailing cap and the anti-reverse cap analog, or the ARCA cap. So in the panel B, we have a very good example or a description of how the co-transcriptional capping happens. Here, let me take an example of the CleanCap AG from TriLink. So the CleanCap AG, when we are using the CleanCap AG, we need to have the template has to be modified in such a way that the template strand contains the TC element right after the T7 promoter. This is required because it base pairs with the AG of the CleanCap and then serves as a primer in a PCR reaction for the transcription reaction. As you can see, since the initiation of the transcription begins with capping, most of the mRNA generated with a co-transcriptional capping mechanism carries the cap, and that way you get a greater than 90% efficiency of capping, which as compared to post-transcriptional capping is about 70%. And due to this difference, co-transcriptional capping has better translational efficiency, longer half-life, and also has boosted and extended protein expression. Moving on, the other two structural elements are the open reading frame and the poly A tail. The open reading frame is the one which encodes for the protein of interest, and this sequence needs to be codon optimized. And this is necessary, A, for translation efficiency, and B, for mRNA abundance. Translation efficiency is achieved by increasing the GC content, eliminating rare tRNA codons from the sequence, as well as using more frequent and highly abundant codons in the sequence. The mRNA abundance can be increased by increasing base pairing and complexity of the secondary structures of this region. Secondly, by removing sequences which act as recognition sites for endonuclease cleavage and chemical degradation. We also need to keep in mind that a COSJAC sequence has to be incorporated at the very beginning of the open reading frame with a start and a stop codon. If not, our protein will not be made. Finally, at the 3' end, we always have a poly A tail because that sort of inhibits or protects the 3' end from deadenylating enzymes, nucleases, and other exonucleases. An ideal length for a poly A tail is about 100. And again, there are two ways of generating a poly A tail. The poly A tail can be encoded in your DNA templates. That way, the mRNA which is made comes off with a poly A sequence at the 3' end, or they can be added enzymatically using poly A polymerases. The disadvantage of having an enzymatic approach is that you get A stretches of variable length, and that way that brings in heterogeneity in your mRNA preparation. With these structural elements in mind, we can start to develop our DNA template. And our DNA template should include a codon optimized open reading frame for better translation, a 5' and 3' regulatory element that has better stability and better translation efficiency. And based on which approach we follow, a poly A tail and a 5' cap either can be incorporated into the DNA template design itself, or can be done after the transcription reaction has happened. Another important thing that needs to be included in your DNA template is a T7 promoter, without which your mRNA will never be made. Now, if you're using self-amplifying mRNA, then you need to include elements such as RNA-dependent RNA polymerases and recognition elements for this enzyme for your self-amplification to happen. And in case if you're using a circular RNA or trying to generate circular RNA, you need to include the circularization junctions and circularization elements in your DNA template, such that after transcription, circularization of your mRNA can happen. Another important thing that needs to be considered is the use of modified nucleotide, and this depends on your use case of the RNA. Commonly used modified nucleotides are methylcytidine, pseudouridine, and N1-methyl pseudouridine. These are commonly used because they reduce your immunostimulatory activity and have a better safety profile. And this is achieved by evading type 1 interferon activity. Once you have your DNA template ready, we can go ahead and see how mRNA can be produced and purified. And this is a very simple cartoon which depicts the whole process. Once your DNA sequence is ready, you get them synthesized and you get them linearized so that the DNA is ready for in vitro runoff transcription. During your in vitro runoff transcription, you have your DNA template, and based on your DNA template, you either include poly A stretches, modified UTPs, and the cap enzyme and perform the reaction. Once the reaction is done, you go ahead and start purifying it. Purification is done using oligonucleotides, which contain a stretch of DTs, and these are specific and bind to the poly A tail, and that way enrich the mRNA. Once the mRNA is eluted from these columns, they can undergo a couple of chromatography runs, and then using a tangential flow, they are concentrated and undergo formulation. Once the formulation has happened, separation of the unformulated molecule is done, and then they are packed and ready for your clinical use. At Sekkei, we have been working with many different mRNA molecules and have entered different stages, and this is one of the ones which is which I want to showcase here. There is a molecule called SPP102, which is about 1,300 nucleotides long. This encodes for a protein which is about 55 kilodaltons in length. We have designed 5' and UTR sequences, and upstream to those, we have used we have DNA elements which bind to the clean cap AG from TriLink, as well as we have included a poly A stretch in our DNA template itself. This template was synthesized, and when we linearized it, we could find a band on an agarose gel at the exact height corresponding to the sequence of the plasmid. We use this DNA to perform an in vitro transcription reaction, and using oligo-DTs, we purified and enriched our mRNA. And when we separated them on a 1% MOX formaldehyde gel, we found the mRNA to be running at 1600 bases, which is between 1 and 2 kb on the gel. We use this mRNA to see if they are active, and in an in vitro translation reaction using rabbit reticulocyte lysate, we could find that when the RNA is present with the cap, of 55 kilodalton protein is made. If the cap is not present, the amount of RNA, the protein made decreases significantly. We use the same mRNA to transfect cells, and when we had transfected cells, we could see that the cells were making protein, and this could be seen on the Western blot. With that, I'd like to come to the end of my talk, and these are the references which I have used. And thank you all for this. Thank you, Mr. Satish, over to you.
Yeah, thank you so much, Dr. Srivastava, for giving us an easy understanding of this complex technology for mRNA production. I think we have quite a few questions, and we shall get back to you in the Q&A session. So once again, I request everyone to please type in your questions in the chat, in the Q&A box only, and not in the chat box, so that we can come back to you with the answers, and we can post it to the panelists and get back to you with the answers. Okay, going forward, a quick introduction of Dr. Anand Ketkar. So he has over 25 years of experience in various biological companies like Biocon, Apple Biologics, and Stelis Biopharma. He, along with his friends, have started Sekkei Bio. So in his strong career, he has proven success in R&D operations from concept to clinical trials to market. He has been driving product strategies, scaling up production, and delivering R&D operational excellence through process, quality, and cost control optimization. So I personally like to discuss a lot of things with Anand, and I'm just awed the knowledge that he has in every aspect of life. He's a guiding force and my guru in many aspects. So over to you, Dr. Anand.
Thank you, Satish. That was a little over the top introduction. Me being your guide is certainly not possible, but thank you very much. So just give me a second. I'll load the presentation. All right. So first of all, thank you very much, Satish, for inviting us for this webinar, and thank you all the participants. There have been some very interesting questions that have been asked. So after Dr. Chatterjee covering the landscape and Srivastava covering the technology itself, I think it is quite apparent that there are several challenges, but with the challenges, there comes the opportunity. And I'm going to talk about it. In a 15-minute seminar, it is just not possible to cover every aspect of the technology and the challenges and the scope. But I'm hoping that in next 15 minutes or so, I'll leave you with enough food for thought. And I'm sure there will be more opportunities to cover each of these topics in details in some other forum. So just to kind of recap, so as you've heard, that mRNA technology has come out from the lab to a large-scale manufacturing and commercialization in a very, very short time. Primarily, Moderna and Pfizer-BioNTech have been instrumental in doing that. There have been some, I mean, CureVac is another company who has been there. But having said that, it just primarily came out because of the pandemic, and there is a lot of things that we still don't understand. So essentially, if you look at IVT mRNA, as early as 2010, it was shown that if you introduce IVT mRNA, then basically there's a significantly higher level of therapeutic protein that can be expressed. It was shown on erythropoietin initially. And then subsequently, people have been trying to adopt mRNA technology for expressing protein using body as a bioreactor. And as you are aware that the MABS is one of the class of therapeutics which is almost 90–95% of modern drugs in recent times have been monoclonal antibodies against various diseases. But if you look at biologics adoption or the MABs adoption well, across the world, it has been fairly low. While we see a lot of numbers on the market in terms of revenues and profits and all of that, if you really look at it, the affordability point of view, there are a number of patients who need the therapy versus how many are being treated with this, there's a very wide gap. And primary driver is the cost. MAB processes are expensive, although there have been a considerable reduction in the cost of manufacturing over the last 5 or 6 years or maybe 10 years or so. It is still not low enough for most of the masses to afford. The process is complex. The PTMs are challenging. You do see aberrant PTMs. The molecules are not very well defined, and so on and so forth. Depending on the antibody, structure, purification needs to be tailored. In some cases, the antibodies are cleared very rapidly, which basically means that the frequency of injections have to be high. And if you see barring 4 or 5 antibodies, most of the antibodies are given as IV and not really as sub-Q, which essentially adds to the complication because the patient has to go to the clinic and then under the supervision, he has to get administered. So there are challenges. And on top of it, of course, the CapEx requirement is very high. If you go for single-use bioreactors, they are at a 2,000 liter scale. And if it is stainless steel reactors, people have now started adopting 5,000 liter and 6,000 liters scale. But it's still very large scale of operation for the number of doses that can be produced per bioreactor. So all of that adds to the availability cost of monoclonal antibodies. And there has been in recent times, there have been a fair bit of attempt to see if monoclonal antibodies can be replaced with mRNA, and rightly so, because mRNA, while it is a very promising technology, if you look at the overall operations of monoclonal mRNA, it requires significantly lower capex. We have done some internal assessments in terms of how much capex would be required for similar number of doses between a MAB and a mRNA, and the gap is really, really wide. It clearly tilts the scale in the favor of mRNA. Essentially, it falls under small manufacturing footprint technologies. So there are advantages, but while having said that, there is a lot to learn. There are several challenges that need to be worked upon and sorted out. And one good thing is, while mRNA technology has helped with the pandemic, it has also shown clearly the areas where there are opportunities, there are current concerns, and there are opportunities that can make this technology accepted widely and globally. So one of the major concerns is immunogenicity of mRNA. When you are developing vaccine or therapeutic product, you don't want the molecule itself to be immunogenic. If you take a unmodified mRNA, it's quite immunogenic, and we'll talk about that in a second. Translatability, that means it has to get translated into the desired protein, and monoclonal antibodies especially form additional or throw additional challenge at you, because you have to have a light chain and a heavy chain, and it's not just the translation of the 2 chains, but they have to come together and form the fully active antibody. So that's additional level. And then the stability of mRNA itself, which has been a fair bit of challenge. All of that ultimately has to have an impact on the dose. So how do you get the mRNA dose right? This is another challenge, and we'll talk about it. There needs to be adequate exposure for the patients. And if you don't, then essentially the whole advantage that mRNA technology promises to provide, that will get nullified. Manufacturing cost, currently it is still high, and people are working across the world to get it down. Transportation and cold chain, again, it is directly proportional to the stability of mRNA. But clearly, if you look at the overall cost structure of any therapy, supply chain is a significant part of this part of the overall cost to the patient. And in case of antibody, clearly the cold chain requirement and deep freezing requirement make it quite challenging to transport the drug from one continent to other continent, like small molecule drugs or other protein drugs. So there is a lot of work that needs to be done. And of course, lastly, the delivery systems. So this is just kind of an overview on the challenges that exist. And there are more challenges, but in the interest of time, I won't be covering everything. So just to kind of recap, immunogenicity, so all the unmodified mRNA are known to be immunogenic. It is recognized by the innate immune system, and the whole cascade, if it is encapsulated in LNP, then it gets internalized by endocytosis, detected by AMAP, and subsequently the whole cascade of inflammatory cascade that starts. If you have a naked single-strand RNA, it is recognized by TLR7 and 8, and then that results in a pro-inflammatory cytokine expression and type 1 IFNs. And this poses a significant challenge, because if one tries to use mRNA as a therapeutic molecule, and if it evokes the immune response, then that's not acceptable. It will become a significant safety concern. Although to some extent the safety concern has been addressed, or the immunogenicity has been addressed by modifying the UTPs, it's still not completely alleviated, and we will, as the field progresses, we will see how the things emerge. Coming to poor stability of mRNA, if you remember when Moderna and Pfizer, they put out their product in the market, the requirement was at -80. It needed to be stored and transported at -80 degrees. And the primary reason is mRNA is a very large molecule, so structurally there is an instability, which is a problem. On top of it, it is also highly sensitive to RNase enzymes, which are freely present everywhere. Wherever there's a skin exposure, or there's exposure to microbes, there will be presence of RNase, which also makes the entire operations very complex. So not only the molecule has to be stable, but operationally you have to take extreme care to ensure that there's no RNase contamination, and that could potentially hopefully stabilize the molecule. Having said that, there's a lot of work needs to be done. Every structural aspect of mRNA is not yet fully understood, which basically means that the field is still evolving, and as we go along, we'll see progress in this area. Now, stability affects the high dose, affects the dose. So essentially, to compensate for loss of potency or loss of translatability, one might require high dose. So there are downside to not just the transportation and storage, but there are direct impact on the product performance as well. So all of these may get addressed by the type and design of mRNA. There's a lot of work that is going on across the world. There's the field is just emerging, and so is the work in Sekkei Bio, where we are also trying to basically look at circular RNA and self-amplifying, and there are other types of RNAs which would be quite helpful, hopefully. And the translatability can be addressed by construct design. Briefly, I think Dr. Srivatsa had touched upon it. Then the question is, question is about getting the dose and exposure right. So I've taken an example of trastuzumab, which is the easiest example we can all relate to it. So trastuzumab IV dose is 440 milligrams, whereas the subQ Hylecta, which is a subQ formulation, the dose is 600 milligrams, which is very high. So now, if you decide to convert trastuzumab protein into an mRNA and use that as a therapy, how would it work? You'll have to have adequate exposure so that the dosing regimen is not very different. And you'll also need to ensure that the dose of mRNA itself is low enough to be cost effective. If you see on the right hand side of this panel, basically Herceptin dose as a protein is 8 milligrams. This was a mice study, I think, which was published in Molecular Therapy, volume 27, number 8. If you look at trastuzumab mRNA, that is just 1/4 of the Herceptin dose. That's a very high dose. If you have to translate into humans, this will translate into almost 100 milligrams of mRNA, which is obviously not going to be commercially viable. So there's a lot of work that needs to be done to ensure that the dosing regimen for mRNA and dosing itself for mRNA is manageable. There are several factors that affect it, and I won't get into the details of all of that. But ultimately, the target would be if you can get 50 or 100 micrograms of mRNA, which would translate into adequate exposure and for long enough duration, then this is the therapy that can actually become viable for replacing monoclonal antibodies into mRNA therapy. Coming to cost, there are several cost drivers. Sorry, I'm just trying to move this. Okay, let me. So the reagents are very expensive. If you look at, then there's multi-step chromatographic purification with low loading capacities in the column. All of that contribute to the high cost. LNPs per se, there's a lot of research that is needed, and people have started focusing on that. But as of now, LNPs getting very consistent quality of LNPs and making sure that they are all RNAs free and they don't contribute to the degradation of RNA. The cost of LNP also contributes quite a bit. And the overall formulation manufacturing process is quite complex. So just to kind of elaborate a little bit on the cost drivers on the RM cost, modified UTPs, enzymes, and cap reagents. These are the three most expensive components of the whole process. If you look at a bill of material, and these would contribute almost to the tune of around 85 to 90% of the overall cost. So which means that there needs to be a lot of innovation and the cost has to come down. Most of the UTPs and cap reagents are under patent by TriLink, and there are some other companies also. So while during pandemic, none of the patents were enforced, going forward, all these patents will come into force, and which will basically create a hindrance to adopt RNA technology. Moving on to purification, a very typical purification flow looks like this. So first you need to make plasmid DNA, you have to purify it. Doing it in a small scale versus doing it at a large scale is a different ballgame altogether. But good thing is when you take DNA, 1 milligram of DNA or 1 unit of DNA translates into 200 to 300 fold amplification. So 1 milligram of DNA will very likely lead to 200 milligrams to 300 milligrams of RNA. So that's a good thing, but you still need to make the DNA, you have to isolate it, purify it in adequate quantities. And then after linearizing, you go through IVT reaction where you make the mRNA, and then it undergoes a few purification steps where affinity purification with oligo-DT affinity purification is one of the steps. And then either it could be AEC, HIC, or sometimes in some cases you might require monolithic columns, then followed by UFDF. So this is sort of a very typical process, and depending on the mRNA, its stability, its behavior, things might have to be looked into. If you look at the loading capacity, it's just 1 to 4 grams of mRNA per liter of resin, which means that if you have a large amount of mRNA, you will have to run multiple cycles, which will indirectly affect the plant occupancy and will have an impact on operational efficiency. You also require at that scale RNase-free water. So you will have to make sure that you generate that RNase-free water. You make sure that everything in the process is free of RNAs and does not introduce RNAs anytime in the system, which makes the whole operation, I would say, a little tedious, and any mishap at the operational floor can basically throw the batch out of the window, which would add significantly to the cost. So there is a considerable scope for improvement. And then there's a LNP formulation and manufacturing process. I won't get into the details. But this is a heavily IP space and considerable innovation that is required on this. So in summary, mRNA is a very promising field. There's a lot of innovation required. There's a lot of focus that's required to make a big impact. The attempts are going on, and hopefully we'll see the results in very short amount of time. And all these factors, essentially, the design, the type of mRNA, the purification, delivery system, all of that will play a significant role in successfully adopting mRNA for therapeutics. So that's kind of the overview on the challenges and opportunities. Thank you very much, everyone, for listening in patiently, and thank you, Satish. Over to you.
Thank you so much, Dr. Anand. So for giving us the landscape of mAbs versus mRNA therapeutics, the challenges and opportunities of mRNA therapeutics, and the cost drivers of mRNA. So we'll get back to you in the Q&A session. So we have received quite a few questions, and we'll get back to you in the end. So in the meantime, it's time to launch our much awaited compendium on Shimadzu solutions for nucleic acid and oligonucleotide analysis. So if anybody wants a copy of this, you can request by typing mRNA in the chat box, and we'll ensure to send a copy of it to you. It's time now to launch the compendium. Yeah, thanks, Mahesh. Sorry for the technical glitch. So whoever wants a copy of this, you can just request by typing mRNA in the chat box, and we'll ensure to send a copy of this. So going to the next speaker, a quick introduction of Ashutosh Scheller. Currently, he's pursuing his doctorate in biotechnology from ICT. He has over 7 years of experience working with Shimadzu Analytical India Private Limited, Mumbai. His profile includes providing Shimadzu solutions for the biopharma market. Shimadzu Analytical India in Mumbai has researchers and scientists and have published and presented a lot of papers on biopharma solutions. Ashutosh and his team have presented papers in many conferences, including the American Society for Mass Spectrometer. Over to you, Ashutosh.
Thank you, Satish, for the nice introduction. Before proceeding, I would just like to thank all our eminent speakers who has very correctly set the stage for the mRNA therapeutics. I will just like to give you a glimpse of the compendium which we have recently shown onto the screen. Give me some time to share my screen. Hope it is, my screen is visible. Satish, can you give me a
Yes.
quick confirmation? Yeah.
Yes, you can go ahead.
After the pandemic, there has been a lot of importance to the nucleic acid therapeutics. Shimadzu, being very innovative in the science and the technology, has came up with multiple applications using the analytical instruments, the chromatographic instruments, mass spectroscopic instrument. Just to give you the glimpse of the compendium, it includes several applications which covers the sequencing of the oligonucleotides using the MALDI-TOF instrument. It's a high resolution mass spectrometer. Next to it, a few of the synthetic compounds or the molecules like the peptide nucleic acids, the exact identification or the analysis of the molecular weight has been introduced into this particular application. We are glad to introduce to our next technology in terms of the UHPLC. We have recently launched the bioinert system, which is very useful considering the complex, considering the analysis of the complex molecules like the oligonucleotides, peptides, proteins, monoclonal antibodies, which has a complex nature, and the analysis becomes challenging when we consider the UHPLC. So with this, we have covered several applications in this compendium. Not only the inertness of the UHPLC, Shimadzu also has a product line. Shimadzu also has several columns in terms of the inertness, bioinert columns, which we used to call it as the oligonucleotide analysis as well as the biomolecule analysis involves several quality checks like the charge variant analysis, the aggregates analysis, the peptide mapping, etc., for which we require specific as well as dedicated columns, which has introduced by the Shimadzu. So just a glimpse to show you in this particular compendium, Shimpac Bio IEX columns. So the ion exchange chromatography, these particular columns are used in the ion exchange chromatography, which offers complete inertness, providing good resolution, a good sensitivity, as well as good chromatography. By using the UV spectrophotometer, the stability studies of the nucleic acids and the relevant drugs are shown in this particular application node with a temperature controller feature which has which is there in our UV spectroscopy. Also, with the inert system, with the bioinert UHPLC system, we have a pH monitor which is placed at the end after the detector, which monitors the pH for each and every sample or data. It gives us a good idea, a fair idea regarding the analysis. Since the pH, most of the bioanalysis, it involves a very crucial or very critical monitoring of the pH, as it has a drastic impact or huge impact on the analysis. So not only this, with the triple quadruple mass spectrometers, which are the gold standard mass spectrometers for the quantitation, for the targeted quantitation of the drugs, any of the drugs, ranging from the small molecules to the large molecules. We also introduced few of the applications for the quantitation of the oligonucleotides using triple quadruples mass spectrometer. Onto the MS, onto the mass spectrometer, Shimadzu has several method packages. Having said this, method packages are nothing but a complete optimized methods, ready to inject methods, which can, wherein you do not have to develop any of the methods, any of the parameters in terms of the chromatography as well as the mass spectrometer. With this, we have dedicated booklets as well, covering all the relevant applications in the biopharmaceutical, vaccines, oligonucleotides, etc. With this, I will just stop sharing the compendium, and I will directly jump onto my presentation. Very good afternoon to all of you. Thank you for participating in this webinar. Having said, thankful to all the eminent speakers, now I will be slightly deviating from the track. So my topic will be to introduce all of you to the analytical equipments, analytical tools for ensuring the quality of mRNA therapeutics. The agenda of my talk will be to introduce you to the basic concepts why analytical tools are required. As you all might be knowing that biomolecules, there are several biological assays ranging from the sequencing, the PCR. There are several bioassays which are still considered as the gold standard for quantifying, for identifying the biomolecules, which is true in case of the oligonucleotides as well. Having said this, regulatory guidelines, they also welcome several orthogonal techniques and analytical tools. The advancement in the analytical tools is so fast that you cannot skip considering the benefits or the advantages which the analytical tools offers. The critical quality attributes on the mRNA therapeutics. mRNA is not a simple molecule considering its complexity, a large molecule, a biomolecule, which tends to have multiple variabilities or variations considering right from the manufacturing to the final formulation stages. So several quality attributes or the test needs to be monitored during each and every step of the mRNA therapeutics. Followed by, I will like to introduce to the few of the chromatographic as well as the spectroscopic applications, which we have covered in the compendium. So just not to introduce only to the application notes, but the basic concept behind using the analytical tools in the mRNA therapeutics. So this is just to show the different classes of the drugs. So basically the drug falls into three different categories: the small molecule drugs or the low molecular weight drugs, which we generally use for treating different diseases. Then the biopharmaceutical to the extreme right end, which are nothing but the monoclonal antibodies, several peptide drugs. In between falls the nucleic acid, which is a recently invented drug, I can say, because there is much focus before the pandemic given to this particular or specific type of the drugs. Considering the x-axis, you can see the drugs which are the small molecule drugs are very easy to which are easily taken by the body, whereas the MABs or the biological drugs, the protein drugs, which are very difficult considering all the different post-translational modification. The nucleic acid drugs are much I can say those are not as complex as the proteins, as well as it is not too simple as compared to the low molecular drugs. In the previous talks, this particular structure of the mRNA has been covered, which consists of different regions for coding the protein. What is important is that considering or having said all these criticalities as well as the complexness of the mRNA therapeutics, a common set of the method is required. An orthogonal method adds value and confidence to the data. Analytical methods are gaining the importance because of its highly specific, selective, and sensitive functions or the features which it offers. Just to introduce you the difference, I mean, why all these critical quality attributes are required for the biomolecules when it comes into the picture. Just to explain you in a very simple way, if we compare a car with an aeroplane, car being a very simple, a very small object in comparison with the aeroplane, it has a less complexity as compared to the aeroplane, which will be having multiple different parts. So similar is applicable to in the chemistry. When you deal with the small molecules, say for example a paracetamol having only a one aromatic benzene ring followed by one or two functional groups. But when it comes to biomolecules, a large molecule which has multiple monomers attached to it and multiple functional groups, the complexity increases. So as a thumb rule, when the size increases, the complexity also increases, which becomes difficult to control the manufacturing as well as maintaining the integrity of these biomolecules or the therapeutics or the biotherapeutics. Having said this, in terms of the size, when size increases, the complexity increases. Not only that, but the production of the biomolecules or the biotherapeutics is also very crucial. It involves multiple stages considering the production of the mRNA therapeutics. It starts from the genetic engineering, wherein the DNA is entered into the plasmid, into the host cell, and then it is allowed to multiply or amplify. The mRNA is then produced in a cell-free system via the in vitro transcription. The mRNA is enzymatically capped at the 5' end, and the 3' end of the mRNA is capped with the poly A tail. The mRNA drug substance is then purified and formulated to make the final drug product. So there are many steps which involves or which adds the complexity to the molecule. The quality of the mRNA drug substance is determined by the design, development, and the specification. So multiple aspects are involved in determining the quality of the final mRNA therapeutics. So this is the table which I took from the draft guidelines, which are defined by the USP. This defines the several aspects or the quality attributes which needs to be checked or needs to be kept into control when considering the production, manufacturing, as well as the final formulation of the mRNA therapeutics. Most of the methods which are involved, you can see there, these are the biological assays involving the sequencing, the next generation sequencing, the PCR, the RT-PCR, gel electrophoresis, different electrophoresis techniques, immunoblot, etc. What I will be introducing you in my upcoming slides will be the orthogonal techniques, like the high resolution mass spectrometers, the MARDI, matrix assisted laser desorption ionization time of flight, as well as the QTOF, which is a hybrid mass analyzer. The first mass analyzer is a quadrupole, and the another second mass analyzer is a time of flight, followed by few of the chromatographic assays, HPLC, UHPLC, different modes of the HPLCs, and finally the LC-MS. When I say MS, it's the mass analyzer, which is a triple quadruple mass spectrometer. So as I told you, orthogonal techniques are always welcomed by the regulatory guidelines, as it adds the values and ensures the it adds the values and the reliability to the data. So to begin with the first test or the quality attributes of the mRNA therapeutics, identity. As we all know that mRNA encodes the genetic information from the DNA. A small change in the mRNA sequence, even one change in the nucleotide, it produces completely different protein. Identification of the nucleic acid sequence is crucial in the mRNA therapeutics. So sequencing is very important when it as it is a part of the characterization as well as in the very initial stages of the drug development. Mass spectrometry for characterization. Generally, when it comes to the analytical platform, the high resolution mass spectrometers are used for the characterization. MALDI-TOF as well as the QTOF, these are the two instruments which are very well utilized for identifying the exact molecular mass of the mRNA therapeutic as well as identifying the sequence of the mRNA. So molecular weight determination is done either by QTOF, which is a hybrid mass analyzer, as well as the MALDI-TOF. So this schematics I have shown you of the QTOF. So this is the QTOF, wherein this is the ionization source. After the introduction of the ions into the quadrupoles, those are fragmented and it enters into the flight tube. So the separation, the resolution of the different molecular ions takes place on the basis of the time. So after analyzing at the detector in the software, we get the ESI mass spectrum, which is a bit complex, considering the complexity of the compounds as well as the function as well as the multiple functional groups which are the part of the mRNA or a biomolecule. So identifying or interpreting the molecular weight with this particular spectra becomes very difficult, considering the multiple charges which the huge or the large mRNA molecule acquires. Deconvoluting the spectra gives you the exact molecular weight of the mRNA therapeutics. So here, after deconvoluting the spectra, the mass which is obtained is 6431.72, which is accurate mass. And if you compare with the observed mass and the calculated mass, the difference is 0.02, which ensures or which shows the capability of the QTOF to determine the exact molecular weight of the mRNA. The another aspect of the characterization is the sequencing. This diagram or this picture is of the MALDI-TOF, wherein you have to load the sample onto the plate, wherein the laser beam will be focused. And this is a linear TOF, where after focusing the beam of the laser, the ions are extracted into the flight tube, and the separation takes place again on the basis of the time. Larger molecule takes a longer time to travel to the detector, whereas the smaller molecule it takes less time to travel to the detector. Now, a very innovative technique of in-source fragmentation, which is known as the ISD, which is ion source decay, is used. It depends on optimization of the matrix which is added during the ionization process. There are specific cleavage sites onto the mRNA, considering the backbone of the mRNA. So with this, we get a sequence. Those specific sites are cleaved, and different spectra are observed or monitored onto the mass spectrum, which gives the sequence of the mRNA or the any of the oligonucleotide. One limitation I can say in this particular technique is this MALDI-TOF sequencing is limited, or it is restricted only to short oligonucleotides having the chain of 50 or 50 mers, not more than that. When the oligonucleotide increases in the size in terms of the mers or the oligonucleotides or the nucleotides which are attached onto it, then the sequencing with the help of the biological assay, DNA sequencing or RT-PCR becomes mandatory or the prominent in that case. RNA content, it helps in determining the titer during the manufacture. So after the mRNA therapeutics are being synthesized, manufactured using either the biological way or the chemical synthesis, you need to define the titer, or you need to identify the titer of the manufacture, the concentration of the drug which is present, which is obtained after the purification. Which it also helps in defining the dosage. So it is not only after the purification, but after the end when you fill the therapeutics into the formulation vials, the concentration also needs to be identified. A very simple technique to identify the nucleic acid concentration is the UV spectroscopy. So in the compendium, this particular application has been published, wherein we have used the UV spectrophotometer 1900i from Shimadzu. The method is used to calculate again the RNA concentration. The absorbance is collected at 260 nanometer and 280 nanometer, and the absorbance is the working principle is based on the Beer and Lambert's law. The ratio at 260 and 280 nanometer is taken, and the value of 1.8 to 2.1, it indicates the highly purified mRNA. So the acceptance criteria to consider the purity of the mRNA lies between 1.8 to 2.1. Then comes the integrity. After the RNA is manufactured, it is processed. And after purification, we need to check the integrity. As we all know that there will be the possibility will be there that there will be many impurities or the process related impurities which will be present in the formulation. So there are multiple ways to check the integrity of the RNA. Basically, all the different chromatographic assays are used to check the integrity. So two of the widely used assays, the modes of the chromatography are size exclusion chromatography and ion exchange chromatography. Size exclusion chromatography depends, the separation depends on the molecular size, the pores available, different size of the pores available in the column. The one which elutes very early is it's an indication that the molecular weight is high in comparison to the rest of the molecules. So with different pore sizes present inside the columns or available inside the columns, you can have a separation. So this particular mode of chromatography can be applied to different compounds differing in the molecular weight ranges, if you properly or if you wisely select all these different columns of the different pore sizes. Another one is the ion exchange chromatography. As we all know that an oligonucleotide is an ionic compound. It's a polar. So the separation can be done on the basis of the ion exchange chromatography. Short chain oligonucleotides are separated with this particular ion exchange chromatography, not the long one. The impurities like the protecting groups and the degradants can be very well separated. So you can see in this particular application, which is again published in the compendium, you can see the protecting groups, they elute first, followed by the shorter length of oligonucleotides, which are again considered as the impurity, followed by the separation of all the 5 different oligonucleotides, which differs in the length. So the length of the oligonucleotides differs from 16 to 20, and using the ion exchange chromatography and ion exchange column, those are very well separated onto the HPLC. Another mode of chromatography, which is again very widely used, is the reverse phase chromatography. Reverse phase chromatography, it is the stationary phase is non-polar and the mobile phase is polar. However, when you use the reverse phase chromatography, you can use the reverse phase chromatography into 3 different submodes. When you want to separate the oligonucleotides, ion pair chromatography, it is nothing but you have to mask the C18 chemistry of the stationary phase with the ion exchangers, and then helic, which is again used for separating the polar compounds. Oligonucleotides being polar compound has to be written onto the column, depending on the differences in the size and the of the different oligonucleotides. So one of the application wherein we have used C18 chromatography or C18 chemistry, and this is nothing but the ion pair reverse phase chromatography. So mobile phase, it has one ion pairing reagent which has been added to the in the mobile phase, and then the separation, you can see this particular big compound is the oligonucleotide of our interest, and the rest of the small degradants or the impurities which are very well separated. Post to this, having said or having discussed all the different modes of the chromatography, as I rightly told in the beginning, that when the size increases, the complexity also increases. So there are many challenges associated with the reverse phase chromatography or the ion exchange chromatography or any of the chromatographic acids. So the nucleotide along a nucleic acid polymer, it contains a variety of the sites for interaction with the metal ions. So these oligonucleotides, they tend to interact with the metal with the 2 principles, electrostatic interaction as well as the nucleophilic interactions with the purine heterosides. This property leads to adsorption of the nucleic acid or oligonucleotides to the metal surface of the HPLC. So as we all know that HPLC, most of the surfaces of the HPLC are made up of stainless steel material. And if you perform the analysis of nucleotides or any of the nucleic acids using HPLC, those nucleic acid tends to get adsorbed onto the metal surfaces of the HPLC, which interferes the analysis, leading to poor chromatography, sensitivity, resolution, and inconsistency issue. What solutions we have from Shimadzu is we have introduced a bioinert system which is completely inert to all the molecules towards the biomolecules, meaning all the surfaces of the HPLCs are coated with an alloy, as well as all the tubings, as well as the needle which takes the injection. It is of the ceramic material and the PEEK material. With this, one of the application which we have shown again in the booklet is, if you use the HPLC system to separate the nucleotides, you will see a bad chromatography peak shape. A tailing is observed which hampers the resolution. Whereas if you use the inert chromatography with bioinert columns, the sensitivity in comparison to these chromatograms, you can see an increased sensitivity. The smaller peaks or the smaller components of the concentration of the compounds which are present in the samples and are very well detected, and you get a very sharp chromatographic peaks. With this, not only the analytical scale, we are also having a very good solutions for in the preparative scale. So after the synthesis, the oligonucleotides are purified. Those can be purified using Nexera preparative system. So pure fractions can be collected. There are different scales at which we have the different columns, the pump size, as well as the fraction collectors. Very importantly, the mRNA capping efficiency study. mRNA ideally has to be capped to provide or to avoid the degradation once it enters the human body. So the cap is nothing but it's a dinucleotide which is added to the 5' end of the nucleotide or mRNA, and 3' end is covered by the poly A tails. So for this particular application, to study the efficiency of capping after producing the mRNA, we have used reverse phase chromatography with LC-MS-MS technique, which is nothing but a triple quadruple mass spectrometer. So in this particular application, what we have done is that the mRNA, which is capped, we cleaved the cap of the mRNA with the nuclease enzyme. The caps are cleaved and released after the digestion. The sample is filtered and it is desalted, which is then analyzed onto the LC-MS 8045. And on LC-MS 8045, we used the MRM mode, which is nothing but a multi-reaction monitoring mode, wherein we targeted the precursor ion of the cap, which is a dinucleotide, in the collision cell. The cap was fragmented. and a specific product ion of a cap was identified or it was detected in the quadrupole 3. So this is a basic skeleton of a triple quadrupole, wherein a precursor ion is selected, it is fragmented, and a specific or signature product ion of the selected precursor ion was detected, which gives a high selectivity as well as the specificity to the analysis. So these were the results of the chromatograms, wherein we studied the selectivity of this particular method in a way that the blank sample, which is a naked mRNA without cap, we perform the digestion. As we know that nucleus, it identifies the specific cleavage site onto the mRNA, and then it cleaves. So we performed the cleavage of mRNA, a naked mRNA without cap, and the sample we injected onto LCMS, and we checked the retention time as the response at the retention time of the standard sample. The response was, we didn't got any interference, which proved the selectivity of this method. Followed by, we acquired a linear calibration. We ran the linear calibration standards across a different range, and the concentration of the caps in the digested samples were extrapolated using external standard method of quantitation. So this is how we developed a method to check the capping efficiency of the mRNA. And this method was developed mutually with one of our customers. This is my last slide of my presentation. Having discussed all the analytical tools which are used in the mRNA therapeutics analysis, we cannot completely rely onto the analytical tools, since the biological assays, which are still considered as the gold standard assays, they have few certain advantages and disadvantages. All the analytical techniques, the different techniques, the biological techniques as well as the analytical techniques must go hand in hand. Not only this, considering the regulations, the different regulations, GLP, GMP, the other compliances as well, all must go hand in hand to ensure a good quality or high quality of the mRNA therapeutics. So this ends my presentation. Thank you for your time and attention. Over to you, Satish.
Yeah. Thank you, Asutosh. And all the panelists, we have received a lot of appreciation from our viewers. So they've been sending me messages for explaining mRNA therapeutics and the analytical techniques in a very simple way, and that too in a very short time. So this webinar is brought to you by Spinco Biotech, India's leading analytical instrumentation company, which was started in 1981. True to its tagline, Spinco is bringing technology and enabling science for the past 41 years. So straight away, let us go into the Q&A session. So we have received a lot of questions, some of them offline as well as some which has been typed in the Q&A box. So I'll start with the first one. So what are the advantages of the newer forms of mRNA, self-amplifying and circular? So this probably Dr. Mona Lisa, probably you can answer this.
Yeah, I can take that. So there are many advantages. So if you can, as it has been mentioned, that the linear mRNA, when you put a certain number of linear mRNA into our body, they don't replicate. They translate, and after they are pretty short-lived, the body clears it, and then the protein is no longer there. So one advantage of self-amplifying, if it continues to make copies of itself, you can have longer duration of protein expression. That's one of them. The advantages of circular are many. Circular mRNA, interestingly, is cap-independent expression. It is cost-effective. It is considered, there is early preliminary data, to be very careful, is not seen as immunogenic by our innate immune system, and it is less labile. There is data that shows that it is better shelf life stability as well as better metabolic stability. So the both, as again, depending on the use case and the design attribute that is needed for your product, I think mRNA has to get designed. If you want a longer duration, you want a temperature stability, so you will have to pick and choose different design elements for addressing the kind of use you have.
Yeah. So the next question is, mRNA therapeutics requires efficient and safe delivery. So what are the commonly used delivery systems? This is open for anybody.
I can take that question. So as of now, if you look at the field, there are limited examples. So what people have tried are LNPs. Typically, mRNA is first stabilized using cationic lipids, and then basically self-assembling lipid nanoparticles are what is used, what is preferred. There's a lot of thrust now where people are trying to look at other materials as a delivery mechanism, because LNPs have availability of lipids for nanoparticle formulation is quite challenging at this point of time. When the pandemic happened, there were very few companies who could supply the LNPs to Pfizer and Moderna, which essentially limited their initial capability to deliver that many number of doses. So right now it's still lipid nanoparticles, but other materials will soon emerge.
Okay.
I hope that answers your question.
Yeah. So going forward, so where are the major opportunities for disruption in the mRNA manufacturing process? What are some of the critical bottlenecks to be overcome? So this,
sorry, come again.
Yeah, once again, where are the major opportunities for disruption in the mRNA manufacturing process, and what are some of the critical bottlenecks to overcome? Probably Dr. Srivatsa can answer this one.
Right. So the mRNA process, the biggest challenge is in the cost. Right now, the cost comes from the caps and the modified UTPs. So one of the ways of eliminating that is to stop using them. So maybe, as Mona mentioned, moving towards a circular RNA variant, wherein you don't have a utilization of the cap, becomes the best way of reducing your costs, and that way the production becomes easier. The other thing is that linear mRNA molecules have a poly A tail, and so you will have to go through multiple steps of purification. So if you have a circular RNA molecule, you can do a purification using a couple of analytical techniques and get away with pure and enriched circular components, which can be directly put into lipid nanoparticles and then tested. So that probably those are the two ways I could think of right now to reduce the production costs.
Right.
I can probably answer the same question. There are many other elements as well. So I think fundamentally you have to understand, one must understand that the cost drivers are clearly the BOM, which is basically bill of material. So cost of material utilized per unit conversion of raw material to product, that drives the cost. And I think this was discussed in my presentation. And the second one is about the plant occupancy, throughput from the plant. The faster this works, the better it is. So these are the main components. Srivatsa, I think, mentioned about the specific steps, but so those are beyond those, there are many other elements that will have to be kept in mind. At this point of time, if the UTRs can be made cheaper, cap can be made cheaper, modified UTPs can be made cheaper, all of that will play a significant role. And then, of course, if the process can be made more efficient.
Yeah.
Okay.
So I think
sorry, just one more thing. Even the method of manufacturing mRNA, the construct design can have an impact on the cost.
So, and going forward, there's one more question. I think this is for Ashutosh. So which mass spectrometer is preferred for oligoanalytics? So either MALDI or triple quad, and for the front end, whether an inert HPLC or a regular HPLC?
Basically, for the characterization at the very beginning stage, where I already included in a few of my slides, where you want to identify the sequence, you want to monitor the sequence, or if you want to identify the exact molecular mass. In that case, the high resolution mass spectrometers like MALDI and QTOF are required. Once it comes into the latter stage, wherein the integrity, I mean the mRNA, the molecular weight of the integrated mRNA, and if you want to quantify it, and further if you want to perform any pharmacokinetics assays, therein you can use the triple quadruple mass spectrometer. Considering coming to the front end, the inert system will be very well preferred because most of the oligonucleotides take it DNA or RNA or the nucleic acids, monomers. Those have high absorption tendency to the metal surfaces. So conventional HPLC will not work in many of the cases wherein you require more saturation time to saturate your conventional HPLC. Again, that I will say it is a temporary solution. So yes, bioinert system is a complete solution in terms of the biocompatibility, wherein the surfaces, the metal surfaces are coated with the alloy, and you can use, you have a provision, you have the flexibility to use the high concentration of the buffers of high pH, as well as the bioinert parts, which are made up of PEEK as well as ceramic. It won't give you any absorption. So yes, the bioinert system coupled with any of the mass analyzer as needed, as per the requirement.
Sure. Yeah, so we have another question from one Mr. Akash Chauresia. So he asks, what is the IP landscape of mRNA-based products? I think this Dr. Monal.
Yeah, I think it's a very general question, but Akash, I'll give you. I mean, we have been mapping the IP landscape. So I'm telling you from first-hand experience, the landscape is very, very competitive. Not just from the product point of view. There are technology landscape, method landscape, technology IP, method IP, use IP, delivery IP, and they're very interlinked, right? Then there are IP of every element of the design of mRNA, right? So you will see that it is a very contested space. And even if we just take, because at Sekkei, we are very interested in circular RNA, and we have been looking at which is a relatively newest field, I would say, from all forms of mRNA. Even there is an exponential level of filings that are happening in all categories. And yet, I would say the field is new. So there is a lot to innovate yet. So giving you a very straightforward response is difficult, to be very fair. I don't think it's feasible in this time frame.
Sure. Thanks. So there's another question from one Mr. Vinod Pantula. So for which category mRNA is more suitable, prophylactic or therapeutic vaccines?
I can start and maybe others can chime in. Prophylaxis, definitely, especially vaccines, because the good thing about prophylaxis is you're asking the immune system to do everything, right? Prophylaxis is when you have not been infected, and you want the body to be ready and protected before the infectious agent comes in. So that makes a lot of sense. In the case of therapeutic vaccine, you have to be careful. If you're talking about therapeutic vaccines, in case of oncology, there has been a lot of attempt where you're actually using immune system to, target your tumors. So it's a little bit of different use case. You're actually driving immune system to now go recognize the tumors and mount a strong immune system and enabling treatment through that. There are lots of challenges in that space. Tumor types are different, distribution is different, and somebody brought it out in one of the questions. And the field is more and more evolved towards more personalized treatment, where you take the specific person's or the patient's tumor type, look at their tumor antigen expression, and mount a response. Having a generalized therapy going to be difficult. I'm not going to say it's very easy. The third space you will see is where a lot of antiviral therapeutic vaccines are coming. Again, I would say you'll have to wait and see how that space opens up. So at this moment, prophylaxis is more, has proof of concept with respect to approved products. Therapeutic, I think we still have to get approved products. And I welcome anybody else adding.
So anybody else wants to speak, or we can go into the next question. So there's one Mr. Muthuswamy Ramakrishnan. He asks, how many RNA modification identified to date? Last year it was 172 RNA modifications, and how many modifications can be used in medical applications? Anybody?
So I think we should clarify the question. Are you asking how many modified nucleosides have been used in an approved product? Or are you asking, are modifications feasible? You yourself have answered there are that many modifications feasible. We have only three products that are approved. We have only pseudouridine and few modifications that are seen into those products. I hope we understand your question correctly. It's.
So in case if you are, if somebody wants to come online and then want to ask some more questions, you're most welcome. You can just raise your hands and we will take your questions live as well. So going forward, so there's a question from one Mr. Bipin Singh. How challenging is to deliver naked mRNA at specific location in the cell tissue? Can you explain what strategy is used to make them target specific?
I think I responded to that question. So it's a good question. This is one of the field which is quite difficult, is targeting of mRNA. Let me take this. You have asked 3 questions in that specific question. So one is tissue-specific targeting or organ-specific targeting. What is known as mRNA majorly is when you give IM injection, it majorly goes to the liver. That's where you will see highest concentration. It goes to the kidney, and you can see systemic levels too. Initially, there was not so much of targeting work that was done. People just gave it and see will have the PD effect, desired PD effect or not. But now there are formulations being developed that claim that they can do organ-specific delivery. Still very early work. With respect to cell type specific delivery, I'm really honestly say I have not come across a lot of cell type specific delivery. What can be done is by design within a cell, you can decide if I want my protein to, be inside the cell, or it should be transmembrane, or it should secrete out, depending on what is the kind of, again, indication you are addressing and what kind of immune system immune response you want to mount, right? You can make that design, but very specific cell type, not aware of it.
So I can perhaps add a little bit to that. So as Dr. Chatterjee mentioned, that there are not too many attempts or too many things. One thing is very clear, that if you have 100 nanometer particle size, then they tend to accumulate in the kidneys and liver. And as the particle size increases, that's when they start becoming more and more non-specific. So as much as I understand, the information is limited. There are some isolated attempts of using modified carbon nanotubes for mRNA delivery. There's some research going on alternative lipid systems, synthetic lipids. But all of those are general delivery systems. I have not come across anything which is very tissue specific or cell type specific. I mean, this is where we are, which basically that also means that there are tremendous opportunities in the nanoparticle field itself for delivery of mRNA. And if you really look at it, despite LNPs, I think the overall efficiency is 1% or thereabout, below 10% for sure, or below 5% for sure. So there's definitely an opportunity to increase the efficiency and get better results.
Just to reiterate, both Mona Lisa and Anand were talking about LNP encapsulated mRNA. Naked mRNA cannot, as far as I am aware, cannot be delivered specifically to a cell type. Also, as soon as you inject naked mRNA, you get a very strong immune response and the mRNA gets degraded instantaneously. So naked mRNA cannot be delivered specifically to a cell type. It's only encapsulated mRNA.
So I think, so can we take another 2 more questions, like if it's fine? Because there are already a lot of questions here. So probably we'll take 2 questions and the remaining we will send it to you, and then we'll, once you get the answers, then we'll send it to them. So there's one question from Akash Torres, probably specific to most of them. Is there any regulatory guidelines available for repeat-based mRNA product manufacturing testing? And he also goes to say, what is the clinical status of any mRNA-based vaccines for cancer?
Anand, will you take? I can take the clinical status. So many clinical cancer products are being tried in clinical phases. If you just go to clinicaltrials.gov and hit cancer, you will get 640 entries. So definitely cancer-specific mRNA products, which could be either immunotherapy, which is basically cancer vaccines, immunotherapy vaccines, are being tried. Again, as I said, there is not a single approved product yet. So not successful yet. We have to wait and see.
Yeah, and the first part is about specific mRNA lipid nanoparticle specific guidelines. Not that I am aware of as of now, specific to mRNA, but I'm sure they will emerge because the field is just exploding.
Sure. Yeah. So we'll take one last question. So there's one question from Narmada Palnati, probably for Ashutosh. So what is the functional group and packing material used in the Shimpac IX column? Is it hydrophilic porous?
I answered I typed it in the chat box. So basically there are two chemistries, quaternary ammonium as well as the sulfopropyl group. So these two chemistries are available in the IX column, the ion exchange column, for cationic and the anionic exchange.
Okay. Okay, so yeah, I mean, there's a few more questions are pouring in. I think we can take one question from Upendra Naik. So can we introduce the mRNA via cells on the action spot in our body? So anybody, the question I repeat again, can we introduce
Yes,
mRNA via cells on the action spot in our body? It says,
I'm not sure I understood the question clearly.
okay, so we'll go into the next one. How mRNA is purified from cDNA template after IVT? This is from Komal Patra.
mRNA contains poly A tails. So we use a oligonucleotide which has a stretch of T's. So because T and A are complementary to each other, they bind to it and everything else passes off and you enrich the mRNA molecule. Hope that answers the question.
Yeah. So I think, so we have come to the end of this session. So we have received a lot of questions. I'll just pass it on to the panelists. And once we get the answers, we'll get back to you. And for all the people who have requested for the compendium, we'll ensure that we have your email IDs and we'll ensure that you get a copy of this probably in a couple of days. Thank you once again. Thanks all the panelists for being so patient and answering all the questions. Thanks Ashutosh as well. Thanks once again for all the participants. So we look forward to having more sessions like this. Thanks once again. So we'll now close this call. Yeah.
Thank you so much.
Thank you very much.
Yeah.
Thank
Thanks.
you.
Bye.
Bye-bye.
Thank you all. Bye.